Yeah, so I'm going to start just by saying that the work I'm describing today is available as a software package that's available on GitHub. A lot of what I'm going to say today is also applicable to the long-ranger software from 10x. And I'm going to focus on sort of the more technical aspects of analyzing 10x data, trying to identify, characterize structural variants. But if you're interested in the biological story, there's a preprint of this story on bioRxiv, so some of the more biological details are there. So just a little bit of background on structural variants. So these are simply defined as large-scale DNA changes. For example, translocation of this piece of DNA from one chromosome to another, or duplication, or an inversion. And in cancer, we get some really extreme examples of structural variation, including aneuploidy, as well as chromothripsis. So chromothripsis is when a chromosome shatters into many shorter pieces, and then those pieces can get randomly reassembled to form novel chromosomal material. So the only practical approach to identifying structural variation in human genomes has been to use genome sequencing using short reads, and I'm sure you're mostly familiar with that. The key thing to remember here is that we're starting with short genomic fragments on the order of 500 base pairs in length, and then when we map the resulting reads back to the reference genome, we can identify differences between our reference genome and our short reads to allow us to identify variants. So there are a number of challenges for the genome scale characterization of structural variation using these standard short read methods. The first is sort of this fundamental issue of the search space. So if we're looking for single nucleotide variants, basically any position in the genome can either match the reference genome or it can show a mutation. So our search space is proportional to the size of the genome. In contrast, a structural variant can connect any locus in the genome to any other locus in the genome. So the search space then is much larger. It's actually proportional to the square of the size of the genome. So fundamentally a much more difficult problem. Another issue with the short read sequencing is that if we've got multiple breakpoints, it's difficult to relate them if they're distant from one another. And this is important because, as I mentioned before, there's lots of types of complex structural variation where there's multiple breaks in the genomic DNA that are formed either simultaneously or one right after the other. And so this is an important question to be able to answer. So just an example of what this looks like. Let's say we've got the maternal and paternal copy of this chromosome, and we've got two events, the insertion of this red sequence and the inversion of the black sequence resulting in the orange sequence. And so since with standard Illumina, your fragment size are only 500 base pairs, if these two events are more than 500 base pairs apart from one another, we can't tell if they're occurring on opposite copies of the chromosome as shown here, or in cis on the same haplotype. And then the third issue for characterizing structural variation is the issue of genome repetitiveness. And so simply, if we've got this blue sequence that occurs on chromosome 1, an identical copy of it also on chromosome 2, if we get a short read that maps to that matches that blue sequence, it's impossible for us to tell if it derives from the chromosome 1 copy or the chromosome 2 copy. And so obviously, as we get longer and longer distance information, then we can span into unique flanking regions and thereby determine that, for example, this read came from the copy on chromosome 1 and not on chromosome 2, and allow us to identify variation that occurs in those genome repeats. Okay, so obviously we wouldn't be here if I hadn't turned to 10x to characterize structural variation. So you're all familiar with the approach here. I'm just going to emphasize 2 points from the Genomics platform. The first is that we're taking very large genomic DNA fragments. So remember, standard Illumina, we're starting with 500 base pair fragments. Here we're starting with 50 or 100 kilobase pair fragments. And we're then partitioning them into these microfluidic droplets. The second important thing to remember here is that each droplet gets more than one fragment. So for the gem code, the sort of first version of the 10x platform, there were on the order of 100 fragments per droplet, and now with Chromium we get about 10 or so fragments per droplet. Okay, so within those droplets we perform random prime PCR to create shorter fragments templated off of the long DNA. And then we perform this across millions of droplets, release the short DNA fragments, and sequence them with standard Illumina. And so the analysis then looks